a very important topic for undergraduate and postgraduate microbiology students. Bacterial genetics basically deals with DNA organization, heredity, variation, gene transfer, gene mapping, and regulation of gene expression. The easiest way to understand this topic is to divide it into two questions. First one is how do bacteria acquire and rearrange genetic information, and second, how do bacteria control and when how much of a gene product is produced. So let's begin. So most of the bacteria possess a single circular double-stranded DNA which is known as nucleoid or xenophore. They may also contain plasmids, which are usually small circular DNA molecules capable of independent replication. Plasmids may carry useful genes such as antibiotic resistance or special metabolic functions. Now we come to a very important concept, horizontal gene transfer or HGT. The three classical mechanisms are transformation, conjugation and transduction. Remember this simple sequence. Transformation is mediated by naked DNA, conjugation is requires cell to cell contact, and transduction is a process mediated by bacteriophage. So transformation is the uptake of free or naked DNA from the environment by a competent bacterial cell. So the basic process is DNA release, then it binds to the cell, DNA uptaken by the other cell, recombination and expression of a new character. Transformation was historically demonstrated by Frederick Griffith using Streptococcus pneumoniae. Later, Avery, MacLeod and McCarthy demonstrated that DNA was the transforming principle. The important point is transformation requires free DNA, but it does not require direct cell to cell contact. So this is the process showing the phenomena or the events of transformation. Conjugation is basically a DNA transfer through direct contact between bacterial cells. The classical example is the F plasmid of E. coli bacteria. An F plus cell contains the F plasmid and acts as the donor, whereas F minus cell lacks the F plasmid and acts as a recipient. The classical cross is F plus into F minus, or it will produce both the A plus and A plus. During conjugation, the donor establishes contact using a conjugative pilus. DNA transfer begins at ODT and one DNA strand is transferred to the recipient. Both cells then synthesize complementary strands. Now remember two special terms, Hfr cell and F plasmid integrated into the bacterial chromosome. It can transfer chromosomal genes. F plasmid is an incorrectly excised F plasmid carrying some bacterial gene with it. Transduction. Transduction means transfer of bacterial DNA with the help of a bacteriophage. There are two major types: generalized transduction. Potentially any bacterial gene can be transferred because bacterial DNA may be accidentally packaged into a phage particle. Specialized transduction. It is the only specific bacterial genes near the prophage integration site are transferred following inaccurate excision. So the easiest memory trick is generalized is almost any gene, and in case of specialized, specific nearby genes are transferred. Gene mapping. Gene mapping means determining the order and relative positions of genes on the chromosome. In conjugation, the basic principle is closer genes are more likely to be transferred together. In conjugation, interrupted mating experiments can determine the specific gene order. If genes appear in the sequence A, B, C, D, and later genes enter the recipient as mating continues, we can establish their order. In transduction, frequent cotransfer indicates closed linkage. Therefore, cotransduction frequency genes are closer, and high cotransformation frequency genes are closer. Complementation test. A complementation test always tells us whether two mutations affecting the same phenotype are located in the same gene or different genes. If two mutations complement each other and restore the normal phenotype, they are likely present in different genes. If they fail to complement, they are likely mutations in the same gene. So remember, complementation occurs in different genes, no complementation occurs in the same gene. Operon concept. Now let's move to gene regulation. An operon is basically a cluster of functionally related genes under coordinated transcriptional control. The major examples we need to understand are lac operon, tryptophan operon and arabinose operon. Lac operon of E. coli controls the lactose utilization. Its major structural genes are lac Z, which indicate beta galactosidase, lac Y, which is responsible for synthesizing lactose permease, and lac A or thiogalactoside transacetylase. The lac operon is an inducible operon. When lactose is absent, the lac I repressor binds the operator and transcription remains largely off. When lactose is present, allolactose binds the repressor, reducing its ability to bind the operator. So transcription can therefore increase. But there is another important regulator, cap-camp complex. When glucose is low, then cyclic AMP will be increased and cap-camp complex forms. It will initiate the strong transcription. Therefore, the maximum expression of the lac operon occurs when lactose is present and glucose is scarce. Tryptophan operon. The tryptophan operon controls the tryptophan biosynthesis. Unlike the lac operon, it is repressible operon. When tryptophan is abundant, tryptophan plus repressor causes active repressor, formation of active repressor, then it binds with the operator and transcription decreases. Therefore, tryptophan acts as a co-repressor molecule. When tryptophan is scarce, the repressor remains inactive and the biosynthetic genes are expressed. So remember, lac is the inducible plus catabolic and tryptophan is repressible plus anabolic. Attenuation. The tryptophan operon has another elegant regulatory mechanism called attenuation. It depends on the availability of charged RNA, charged tRNA tip and couples translation with transcription. When transcription is abundant, high tryptophan ribosome moves rapidly, terminator forms and transcription stops. When tryptophan is scarce, low tryptophan binds with the ribosome pauses and anti-terminator forms, transcription continues. Thus, high tryptophan genes become off and low tryptophan genes become on. Arabinose operon. The arabinose operon regulates arabinose utilization by the bacteria. Its major structural genes are ARA A, ARA B and ARA D. The key regulatory protein is ARA C. In the absence of arabinose, ARA C contributes to a DNA configuration that inhibits transcription. When arabinose is absent, it binds ARA C and changes its conformation, promoting transcription of the ARA BAD genes. Glucose availability also influences this system through cap-cam mechanism. Catabolite repression. Bacteria generally prefer a readily metabolized carbon source such as glucose. When glucose is abundant, glucose increases, cyclic AMP decreases. So the cyclic CAP, catabolite activator protein activation, will be also decreased. Alternative carbon utilization genes are expressed less efficiently. When glucose is low, cyclic AMP will be increased, then cap-cam complex will be increased and alternative carbon utilization genes are activated. So this phenomena is called catabolite repression. It can produce dioxic growth. When E. coli uses glucose first, it experiences a short lag and then switches to lactose utilization. Riboswitch. Riboswitch is a regulatory RNA element that directly binds a small molecule or metabolite. It generally contains two important regions, aptamer domain, which binds the ligand, and expression platform changes RNA structure and regulates gene expression. A riboswitch can regulate transcription, translation and RNA stability. So remember, riboswitch RNA directly senses a molecule and changes gene expression. Allosteric control. Now let's move from gene regulation to enzyme regulation. In allosteric regulation, a regulatory molecule binds to an allosteric site which is different from the active site. This changes the enzyme's conformation and consequently its activity. An allosteric effector may be positive, increases activity, or negative, decreases the activity. Many allosteric enzymes show cooperative binding and it may produce a sigmoidal substrate velocity curve. Feedback inhibition. Feedback inhibition is basically an important example of metabolic economy. Imagine the pathway A, B, C, D, E. When the E becomes abundant, it inhibits an enzyme involved early in the pathway. Therefore, end product when increases, early enzymes are inhibited, further production decreases. This saves energy and prevents unnecessary accumulation of metabolites. A classic example is isoleucine biosynthesis, where accumulated isoleucine inhibits an early step in its own pathway. So feedback inhibition controls the activity of an existing enzyme and repression controls the synthesis of the enzyme. So isoenzymes, basically it finally let's understand isoenzymes or isoenzyme, which are different molecular forms of enzymes that catalyze the same biochemical reaction but may differ in structure, kinetics or regulation. They allow cells to regulate the same metabolic reaction differently under different conditions. In branched pathways, different isozymes can respond to different end products. This can produce multivalent feedback inhibition, allowing one end product to inhibit its own pathway without unnecessarily stopping the production of another required product. So my dear students, bacterial genetics is not simply about the transfer of DNA, it is about how bacteria acquire genetic information, rearrange it, express it and regulate metabolism efficiently. Understanding this mechanism help us explain adaptation, genetic diversity, metabolic flexibility and the spread of antibiotic resistance. Thank you my dear students for watching this video. Keep learning, keep revising and keep exploring the fascinating world of microbiology.